Hi, in this video, we'll be going through a brief history and technique of patching. We will also be talking about some of the limitations and the impact patching has had on science.
In the 1660s, experiments demonstrated that stimulating a nerve triggered muscle contraction. Galvani supported the electric nature of the nerve impulse and the relationship between stimulus intensity and muscle contraction. He also studied the accumulation of positive and negative charges on the muscle and nerve surfaces. The delay between the application of the electrical stimulus and the muscle contraction was measured by recording the speed of the nerve impulse propagation. These values were in the range of 25 to 40 meters per second. The resting nerve interior was found to be 60 millivolts more negative than the surface. Gilbert Ling and Ralph Gerald were the first to pull glass pipettes to form microelectrodes. Frederick Pratt and John Eisenberg stimulated a single skeletal muscle fiber and took extracellular recordings. The first intercellular experiments used an artificial salt solution to replace part or all of the cytoplasm in squid axons. Voltage clamping was developed in 1939. This technique allowed impedance measurements using extracellular electrodes on axons isolated from squid. It also demonstrated that membrane excitability is determined by passive ion fluxes according to their electrochemical gradients.
Neher and Sackman predicted that regulation of ion channels is responsible for many functions in cells and developed patching as the first method to detect small electrical currents as they pass through a single ion channel. Patch clamping proved to be most effective in excitable cells. It was the first technique to demonstrate the role of ion channels in cell functions. It is an important tool for understanding disease mechanisms and observing the effects of drugs on ion channel activity. Patch clamping also allows for measuring of subthreshold potentials.
Before we start patching, we have to pull the electrode to the right radius, depending on the type of cells, because different types of cells are different radius. So as you can see here, there's a pipette set through a filament which will burn it and pull it at a certain pressure, causing it to break with a certain radius. And you'll see the tip of the pipette form as soon as it breaks, right there. So after we pull the electrode, we insert it under the microscope via the recording electrode. As you can see, the electrode is able to penetrate the tissue of the cells, and the recording electrode connects to the amplifier, which connects to the computer and allows us to record the electrical activity of the cell. As you can see, we are in a current clamp and recording the voltage of the cell. So when you're looking through the microscope, this is something similar to what you would see. As you can see, the electrode towards the right, and if you focus in, you'll see the cell that is about to patch into. As you can see, it's very difficult to see, which makes electrophysiology almost an art. We use this manipulator to control the X, Y, and Z of the recording electrode, which allows us to precisely target one particular cell and record from that particular cell. So along with the electrophysiological experiments, we are also able to do pharmaceutical experiments where we can perfuse drugs into the recording chamber and study the specific actions of a particular neuromodulator, whether it being an agonist or an antagonist for a particular receptor. This allows us to study the particular electrical activity.
When the patch is made, the contents of the pipette diffuse into the cell. The rate of diffusion is slowed to less than 50% at the junction of the pipette and the cell. This is due to the volume of solution remaining constant while being squeezed through a smaller opening. During this diffusion between the pipette and the cell, the contents of the much larger pipette slowly replace the contents of the cell. After about 10 minutes, the contents of the cell are no longer that of the original cell. When a cell is patched, a high-resistance seal is created by the pipette. This seal can cause physical stress on the membrane-cytoskeleton interactions. As a result, ion channels may show increased or decreased mechanosensitivity compared to that of the original cell. Since the cell is damaged once the patch is removed, and drug concentrations can't be changed once a cell is patched, a new cell is required for each point on a dose-response curve. Furthermore, researchers cannot control the contents of the cell and must work with what they are given.
In on-cell patching, the concentration of drugs in the pipette is changed to study ion channel activity under various conditions. Observing channel activity in different drug doses is used to construct a drug dose-response curve. Using inside-out cell patching, scientists can expose the intracellular surface of ion channels to different solutions to observe channel activity under different ligand concentrations. Whole cell patching was developed to record currents over the entire cell membrane. Outside-out patching provides another way for constructing drug dose-response curves where different drug solutions can be tested in the same pipette to measure channel activity.